Hello everyone. In this module, we will discuss about the role of nitric oxide in plant growth and development. Nitric oxide, a gaseous reactive oxygen species, has been identified as a signaling molecule to participate in the various physiological processes in plant growth and development. The learning objectives of this module are an introduction to nitric oxide biosynthesis, role of nitric oxide in plant sexual reproduction, role in root development, role in plant pathogen interactions, the nitric oxide and ROS connection in immune signaling, nitric oxide and antioxidant enzymes, the role of nitric oxide in beneficial plant-microbe interactions, nitric oxide and abiotic stress, nitric oxide, its role in plant pathogen interactions. Nitric oxide is a signaling molecule that mediates many plant responses to biotic and abiotic stresses. In the year 1992, nitric oxide was named Molecule of the Year because of its widespread biological significance, and in the year 1998, Fogot, Murad and Ignarro were awarded the Nobel Prize in Physiology and Medicine. Nitric oxide has a dual function in both regulating growth and development and also mediating plant responses to diverse biotic and abiotic environmental signals. Nitric oxide is now recognized to act as an ubiquitous cell signaling molecule involved in various processes such as root growth, iron uptake and its sequestration, flowering, pollen tube growth, stomatal closure or hormonal signaling. It has also been linked to the plant adaptive responses to pathogenic and symbiotic microorganisms. The major sites of nitric oxide biosynthesis in a plant cell are the protoplast, the chloroplast, the mitochondria and the peroxisomes. The cytoplasm, the cell membrane, the endoplasmic reticulum and the apoplast can also generate nitric oxide in vascular plants. 8 distinct enzymatic and non-enzymatic processes have been recognized which can elaborate nitric oxide in plants. The cytoplasm, the mitochondria, the chloroplast, the peroxisomes and the apoplast are the sites of reductive nitric oxide generation from nitrogen dioxide, that is NO2. The NO2-NO reduction can be catalyzed by assimilatory nitrate reductase NR or the mitochondrial electron transport chain. Deoxygenated heme-containing proteins can also facilitate the reductive nitric oxide generation from NO2. Non-enzymatic NO2-NO reduction can also occur in acidic compartments of the plant tissues. There are various routes of nitric oxide production in plant cells. Reduction of nitrite produces nitric oxide via enzymatic and non-enzymatic routes. Nitric oxide can be synthesized enzymatically from nitrite by nitrate reductase. There are various routes of nitric oxide production in plant cells. Reduction of nitrite produces NO via enzymatic and non-enzymatic routes. Nitric oxide can be synthesized enzymatically from nitrite by nitrate reductase. There is also evidence for L-arginine dependent NO synthase activity in plant cells. Although the protein AT-NOS1 is no longer considered to be a nitric oxide synthase, and no other plant candidate for the role has yet been identified. Evidence also exists for the activity of nitrite NO reductase in roots and for the ability of both chloroplast and mitochondria to convert nitrogen dioxide to NO. Nitric oxide can also be generated by non-enzymatic mechanisms. For example, via chemical reduction of NO2- at acidic pH or by carotenoids in the presence of light, and also at an acidic pH in the presence of a reductant such as ascorbic acid. Nitrate reductase, in addition to its primary nitrate oxidoreductase activity, is capable of reducing NO2 to NO with a lower efficiency. Nitrate reductase mediated NO synthesis is involved in various physiological processes like pathogen defense and stress response. Mitochondria in the green algae and higher plants can use NO2 as an alternative electron acceptor to sustain ATP synthesis under O2 deprivation. The mitochondrial respiratory chain is able to reduce NO2 to nitric oxide at the complex 3, that is cytochrome BC1, and the complex 4, cytochrome C oxidase. This mechanism results in mitochondrial NO generation in plant cells experiencing hypoxia. Plant peroxisomes can also generate NO by NO2 reduction under hypoxic or anoxic conditions. The responsible mechanism may be NO2 NO reducing ability of deoxygenated heme containing proteins in the peroxisome matrix. The enzymatic oxidation of L-arginine to NO and L-citrulline has been identified in the chloroplast and the leaf peroxisomes of the vascular plants as well as in green algae. The oxidation of L-arginine to NO in the chloroplast requires NADPH and is independent from the calcium supply. In the leaf peroxisomes, the L-arginine L-citrulline conversion requires calcium, calmodulin, FAD, that is flavin adenine dinucleotide, FMN, flavin mononucleotide, and NADPH. Polyamines and hydroxylamine can increase the oxidative NO synthesis in plant cells. Nitric oxide was first proposed to be involved on growth and steering by its negative chemotropic effect on lily pollen tube. Roles for nitric oxide and reactive oxygen species, that is ROS, during pollen tube growth have been well established in angiosperms. But there is lack of information regarding the potential signaling roles in the pollen tubes in gymnosperms. The auxin IAA triggers a transient NO accumulation in the roots. In turn, nitric oxide regulates the activation of a MAPK signaling cascade. The auxin IAA triggers a transient NO accumulation in the roots. In turn, NO regulates the activation of a MAPK signaling cascade. This pathway could be mediating both mitotic process and the expression of auxin responsive genes. Nitric oxide can induce adventitious root development in monocot, dicot, and gymnosperm plant species. Many secondary messengers involved in signaling cascades regulated by nitric oxide are implicated in adventitious root development. Two parallel and independent pathways have been described for the role of nitric oxide in induction of adventitious roots. The first proposes the utilization of cGMP through an NO-mediated activation of the enzyme guanylate cyclase. However, the second pathway involves MAPK cascade, which has been reported to trigger the auxin IAA for the transient NO accumulation. Nitric oxide mediates its biological effects in plants facing microbial pathogen attack in various complex signaling pathways shared with reactive oxygen species and the secondary messenger calcium. Nitric oxide produced in plant cells challenged by pathogens, microbe-associated molecular patterns, damage-associated molecular patterns, is integrated in signaling cascades leading to the expression of defense-related genes, production of secondary metabolites, and to the hypersensitive response. The interplays between nitric oxide and reactive oxygen species produced in plant cells challenged by pathogens or derived signals are complex. Two schools of thoughts have been proposed for the crosstalk between NO and ROS. The first one proposes that nitric oxide and ROS positively influence their respective production. Nitric oxide amplifies the oxidative status of cells by inhibiting through S-nitrosylation catalase and ascorbate peroxidase, the two main hydrogen peroxide degrading enzymes. Nitric oxide and hydrogen peroxide act as key essential intermediates in the pathways leading to the expression of ubiquitin ligases and ultimately to the cell death. The ability of nitric oxide and ROS to interfere with the ubiquitination system is further supported by the presence of chaperone-like enzyme CDC48, which is inhibited through S-nitrosylation. In the second school of thought, it has been proposed that NO regulates ROS-induced effects by negatively controlling oxygen and hydrogen peroxide levels via the production of peroxynitrate and through the inhibition of NADPH oxidase by S-nitrosylation. More generally, owing to the ability to inhibit key defense mediators such as NPR1 or SABP3, NO might be a component of the feedback mechanisms controlling the amplitude of defense responses. Nitric oxide is also able to control different biological processes in plants by directly modifying proteins through covalent post-translational modifications, giving rise to nitration, nitrosylation or S-nitrosylation. Nitric oxide is also able to modulate ROS levels by regulating the antioxidant systems. Catalase, one of the chief enzymes involved in degrading hydrogen peroxide produced in peroxisomes, is S-nitrosylated and nitrated, causing both post-translational modifications and a loss of protein activity. Nitric oxide can also react with the heme group of proteins, thus supporting a role for nitric oxide in regulating H2O2 levels. Along with various other signaling molecules like H2O2, nitric oxide plays a key role in the establishment and functioning of rhizobium legume symbiosis as well as mycorrhizal formation, both ecto and endo. In the senescing modules, an increase in ROS and RNS, that is reactive nitrogen species, causing nitro-oxidative stress has been reported, which leads to a reduction in the ability of the symbiotic leg hemoglobins to scavenge oxygen due to modifications mediated by these reactive species. Nitric oxide is produced in the roots of Medicago truncatula when they encounter the exudates of the fungus Gigaspora margarita. Nitric oxide is also involved in the interaction of olive seedling roots with the arbuscular mycorrhizal fungus Rhizophagus irregularis. Nitric oxide has begun to emerge as an important endogenous signaling molecule in the adaptation of plants to abiotic stresses. Researchers have found a role of nitric oxide in a variety of stress responses including drought, salt, heat and cold stress. Removal of nitric oxide has been shown to inhibit ABA-related stomatal responses. NO has also been suggested to activate mitogen activated protein kinase, that is MAPK signaling cascades, which may then drive stomatal closure. To conclude, we can say that nitric oxide combined with other molecules is an important component in the functioning and physiology of plants. In the most nitric oxide regulated processes, plant responses are not activated by nitric oxide alone. However, it is a result of a network of connections between various signaling molecules and pathways. Researchers have been considering the mechanistic details to provide new insights into our understanding of nitric oxide in the context of plant growth and development, plant interaction with beneficial microorganisms like mycorrhiza and rhizobium nodules, as well as defense mechanisms against adverse environmental conditions caused by abiotic and biotic stresses. To summarize, we can say that nitric oxide finds various roles in plant growth and development. However, the intricate mechanisms of its interaction with various biochemical molecules in the plant physiological processes are yet to be elucidated.